Hi, everybody. My name is Sean Hannigan, and I'm going to be presenting on Legionnaire's disease today. I hope you thought of the Romans, but if you haven't today, now you have. Legionnaire's disease is one of the emerging diseases that occurred in the mid to late 20th century. It was first identified at the American Legion Conference in Philadelphia in 1976. For those of you that don't know, the American Legion is the United States veteran nonprofit organization, so they help fund local political events and raise awareness and basically collaborate on behalf of all veterans together at the national level. So unfortunately, out of these 182 cases at this convention, 29 passed away. And that was very new because no one knew what the heck was going on. But it was found that the causative agent was Legionella pneumophila, a gram-negative bacterium. And then shortly after that, the genus Legionella was established 3 years later in 1979. So I just want to talk a little bit about Legionella pneumophila. It is a gram-negative bacterium found in freshwater environments, so lakes, creeks, ponds, you name it. In the case of the American Legion outbreak, it was the man-made water environment that was the reservoir for the bacterium to grow and multiply. But in nature, they exist as intracellular parasites of freshwater protozoa. So that's amoebae and ciliated protozoa. And however, they are opportunistic pathogens when they infect mammalian cells, especially the phagocytic cells of the mammalian, like human immune systems. They grow best between 25 and 42 degrees Celsius. Optimal temperature is 35. So that makes them mesophiles, and which is why they can grow so well in our water systems, because they are designed to grow in temperatures that we like as well. But within the genus of Legionella, there is 48 species comprising 70 serogroups. However, Legionella pneumophila contains 15 serogroups, whereas every other species that was mentioned only contains one serogroup. But in the further addition, the disease, Legionnaire's disease, is only caused by serogroup 1, and that's at least 80% of confirmed cases. So it's pretty clear it's pretty clear what serogroup is the clear cause of Legionnaire's disease most of the time. And then here's some biochemical data. So we got, most of these tests, if you remember, we did in class. But for example, it's catalase positive, it liquefies gelatin, it's oxidase positive in some cases. It's negative for nitrate, negative for urea, negative for carbohydrate. It's grown on buffered charcoal yeast media, and its colonial morphology and utilization of L-cysteine is how it's identified among other species of Legionella and among other bacterial families as well, and genuses. So the Legionnaire's disease is kind of the subset of conditions, or like subset of conditions that one can get. So being infected with Legionella pneumophila leads one to develop legionellosis, which is just a broad term that includes Legionnaire's disease and Pontiac fever. Now, the difference between these two is that, they're caused by the same bacterium, so you would think they have similar results, which they do for the most part. Legionnaire's disease is really, it's a multi-system component that is, it is demonstrative to the immune system, the respiratory system, the muscular system. It affects more than just one system in the body, and it has longer lasting effects, and it can potentially be fatal if not treated properly. especially if you're an at-risk person. Pontiac fever, on the other hand, is more of a lesser degree version of Legionnaire's disease. So if you want to think of it like this, it's a limited self-like, flu-like illness. Usually only, healthy, healthy, healthy adults, in their prime years are most likely to develop Pontiac fever, and they recover pretty well. Legionnaire's disease, generally it affects the older population and the young, and the immunocompromised, and et cetera, et cetera. Like, the same symptoms you've heard for, or the same at-risk factors you've heard for other pneumonia-type diseases. But that being said, Legionnaire pneumophila causes respiratory disease, Legionnaire's, and Pontiac fever when the host inhales aerosolized water containing bacteria, and that's approximately 90% of all Legionellosis cases. And so 8,000 to 18,000 patients are hospitalized in the U.S. annually, and incidence is largely underreported due to several factors, and that includes misdiagnosis, a lack of specific testing, and potential lawsuits. Due to the nature of this bacteria being able to, residing in man-made aquatic systems and, not having the proper maintenance taken care of it, that's why I bring up the lawsuits, because if the hospitals were to start reporting these outbreaks or these developments in disease in their building systems, then that would open them up to litigation, and that would not be fun. So they try not to report that if they do find it. Now, in regards to the misdiagnosis, it's really hard to distinguish Legionnaires from other pneumonia types, based on chest X-ray diffractions, which is our chest X-rays, because that's what they use. That's what they normally use to determine if you have pneumonia or not. And so you can't really just determine that from pneumonia only, from the chest X-ray only. And so, and then again, it's because of the nature of this disease, it's considered a preventable illness, and controlling, eliminating the bacteria in the water reservoirs of our building systems will prevent most cases, which is very true. So next I want to go over some signs and symptoms and the risk factors, some more of the standard stuff. So like the flu and like a lot of other pneumonias and whatnot, there's the main signs and symptoms are fever, headache, muscle pain, non-productive cough, rigors, diarrhea, and dyspnea. Nothing too complicated there. Risk factors: increased age, chronic lung disease, lung cancer, smoking, diabetes, being of the male sex, and the immunocompromised. And if you can think back to the American Legion, that photo I had of the older veterans, they fit all these risk factors pretty well, which is why there was such a big outbreak at that convention. And so they assign most of these symptoms based off what happened at the convention. And, over time, it just, men are twice as more likely over 50 to contract the disease and actually show symptoms of it. And because it's a respiratory disease, smoking and other issues that come from smoking and other lung issues will have an effect on that as well. Sorry about that. So reservoirs and transmission. Like I said, most cases are attributed to human-made aquatic-like environments, although they can be found in nature within as the parasites within protozoa. One example could be within, decorative fountains, as you can see right here in this little diagram that I have, that demonstrates the different areas where Legionella can develop in a private water system. So these aquatic-like environments include central air conditioning systems, cooling towers, humidifiers, whirlpool spas, outdoor fountains, showers, hot tubs, anything else you can think of that uses a main water supply uh, to get water in and out of the mechanism, so to speak. So because of that, you know, we know that biofilms can develop in all of these systems just due to poor sanitation and maintenance support over time, uh, and that it promotes bacterial growth within these systems, and especially with the biofilms, it makes it a lot more difficult to be able to kill these bacteria because they are sheltered and they have more access to the nutrients that they need within these biofilms. But I should point out that transmission does not occur from person to person, which is a good thing. Each person must come into contact with the bacteria individually through aerosolized water or the inhalation or consumption of contaminated water. So I want to talk about pathogenesis for the minute because it's very similar between how they work in amoebae versus in mammalian phagocytic cells, so macrophages. So in both cases, the bacterium enters the cell, and there when it gets inside, it replicates intracellularly. It has some intracellular motility, as you can see here, and there's two types. There's a replicative form and a non-replicative form. The replicative form is smaller, more compact, and has more flagella compared to the non-replicative form. And it basically the bacterium enter the cell by a form of phagocytosis called coiling phagocytosis, and that vesicle fuses with the lysosome, and these bacterium they are able to inhibit the enzymes and the lysozymes from functioning correctly, so they don't get destroyed after fusion with the lysosome. And so once they have developed enough and they are able to start moving around intracellularly, they release from macrophages and from amoeba by two methods, either apoptosis or necrosis. And there's not much more to that. It's pretty standard. They don't remain within the host for very long. They get in, do their job, and get out. For diagnosis, right now, traditionally there have been like three established methods. There's culture of respiratory secretions, urine antigen detection, and serologic comparison of the blood samples taken 3 to 6 weeks apart. So the culture method is the most specific, but their sample, mucus samples that you cough up decay very quickly, so that's not viable. Urine antigen, it's quick and reliable, however, and it gives immediate results, but it only identifies serogroup 1 as of right now. So it misses 40% of potential cases in this respect, but it is the most common diagnostic method. And then the serologic comparison is effective, but it takes long for a while. Treatment, the main traditionally has been erythromycin for many years, but now it's azithromycin and fluoroquinones, specifically levofloxacin, are preferred now because they have less side effects. Pretty standard, nothing else really other than antibiotics. Now, nosocomial infections. So hospitals are a very big proportion of the Legionnaire's disease cases because of large number of at-risk persons, the plumbing systems, and the water temperatures. So some methods that have been, to prevent bacterial growth are the use of monochloramine as biocide instead of chlorine. It works a little bit better than chlorine, and thermostatic mixing valves to prevent scalding of the patients with the water supply. And they're used in municipal water systems as well, which has dramatically reduced potential outbreaks. But yeah, that's it. Thank you